Welcome back, everybody. Our next presentation is by Ben Zhou and Sam Wang from, first of all, Oklahoma School of Science and Math in Oklahoma City, Oklahoma. And Sam is from the Norman North High School of Norman, Oklahoma. And their presentation is titled From Wastewater to Hydrogen, Microbial Community Structure for Electrohydrogenesis. Welcome.
Good morning, ladies and gentlemen. My name is Sam Wang, and this is my teammate Ben Zhou. Today, we feel very honored to be at George Washington University to present to you our project, which is about converting wastewater into hydrogen. I will be covering the background information to our project, and my partner will go on to cover the experimental parts. Now, today's society faces a huge energy crisis. We can look at the United States for an example. In the United States, 98% of all energy for transportation is derived from petroleum. Now, this poses a large problem because, one, petroleum production is expected to peak within the next 10 to 30 years, but then decline. And two, the consumption of fossil fuels such as petroleum leads to heavy air pollution and carbon dioxide emissions. Thus, if society cannot decrease its reliance on fossil fuels, then in the future we will see increased transportation costs and a damaged environment. But thankfully, there's a solution to the energy crisis, and this solution comes from bioenergy. Now, bioenergy is defined as any kind of energy derived from biomass, and three examples of bioenergy are bioethanol, biodiesel, and biohydrogen. Now, our study focuses on biohydrogen because it is clean, efficient, renewable, has no carbon dioxide emissions, and has no toxic byproducts. In fact, this is an example of a car that can actually run on hydrogen, proving that it is a viable source of alternative energy for the future. The municipal sector in the United States also produces significant amounts of wastewater. And this wastewater, which is from places such as bathrooms and kitchens, has in it a variety of organic matter that must be removed before the wastewater can be reused. This organic matter is also a rich source for hydrogen production. Thus, our study focuses on converting this organic matter into hydrogen, in turn treating wastewater and expanding our alternative energy resources at the same time. We can look, for example, at the Netherlands. The Netherlands produces about 1.1 million gallons of wastewater every year. If they were able to change this wastewater into hydrogen, the hydrogen produced would be enough to sustain about 10% of all of their energy needs. Now, the current methods for hydrogen generation includes fermentation, and fermentation is widely considered to be the most efficient method for hydrogen production. The most commonly used substrate for fermentation is glucose because of its availability. Now, if we look at glucose fermentation, we see that if glucose is fully oxidized to carbon dioxide, we could theoretically get 12 moles of hydrogen per mole of glucose. However, with fermentation, the maximum theoretical yield is only 4 moles of hydrogen per mole of glucose. However, due to major inefficiencies within the glucose fermentation process, we can only realistically get about 2 to 3 moles of hydrogen per mole of glucose. So the major question is, how can we recover the remaining 8 to 10 moles of hydrogen? The solution comes to us from microbial electrohydrogenesis cells. Here we see that biomass in wastewater, such as those from kitchens and toilets, is placed into a fermentation tank. And after initial fermentation, we get about 2 to 3 moles of hydrogen. Then the end products from this fermentation reaction, in this case acetate, are placed within the microbial electrohydrogenesis cells where they are oxidized to generate an additional 6 to 8 moles of hydrogen, giving us a grand total of 8 to 11 moles of hydrogen, which is 3 to 4 times the yield from fermentation alone. Now, this is how a microbial electrohydrogenesis cell works. There's an anode chamber and a cathode chamber. When wastewater is pumped into the anode chamber, it brings with it bacteria and organic matter. The bacteria compete to attach to the anode, and when the organic matter reaches the anode, the bacteria will oxidize it, which simultaneously causes an electron to travel through an external circuit to the cathode and a hydrogen proton to travel through a semi-permeable membrane to the cathode. Finally, a small external voltage is applied to the cathode that fuses the proton and the electron into hydrogen gas. However, there are some problems within microbial electrohydrogenesis cell technology. And among these problems are low hydrogen yields, a high variation in experimental conditions, and an unpredictability of reactor stability and performance. Thus, the real challenge is that we lack the effective microbial communities for efficient hydrogen production.
Thank you, Sam. We had two main objectives for our experiment. Our first objective was to develop an effective community of bacteria that could efficiently produce hydrogen from wastewater. Our second objective was to better understand how differences in microbial community structure affect the hydrogen production in our reactors. We had three hypotheses for our experiment. Our first hypothesis was that the hydrogen production yields of each reactor will be different among them. Our second hypothesis was that the microbial community composition and structure will also be different among our reactors. Our third hypothesis was the differences in hydrogen production yields could be due to differences in the microbial community structures in our reactors. This was how our experiment was set up. We had 5 reactors labeled A, B, C, D, and E, each inoculated with the same wastewater, fed glucose as a sole carbon source, under the same operational conditions of pH 7, 25 degrees Celsius, and operated them for 50 days. For every 2 days, we collected the approximately 26 milliliter solution inside the reactor for processes such as DNA extraction. Samples from 3 of the days were used as biological replicates for geochip analysis. What exactly is geochip? Geochip is essentially a technology, it's a glass slide with over 30,000 DNA probes that are able to target genes responsible for various biochemical processes such as carbon, nitrogen, sulfur, phosphorus cyclings, organic contaminant degradation, and metal resistance. Geochip was a crucial tool in our study, and it won the prestigious R&D 100 Award in 2009. With Geochip, we were able to analyze the community structures in our reactors, and this is how we did it. First, we extracted the DNA from a cell pellet of our sample and then amplified it or increased the amount of DNA. After DNA amplification, we labeled the DNA with cyanide dye in order to see it and then hybridized it with Geochip in hybridization. After hybridization, we washed the array and image processed it with a scanner and then implemented various statistical methods to analyze the microarray data in data analysis. In order to measure how much gas was produced by each reactor, gas chromatography was used, or separation of gases, focusing mainly on the production of hydrogen, methane, and carbon dioxide. Here are the results for the gas chromatography analysis. This is a bar graph of the hydrogen production yields of each reactor. As you can see, each reactor produced different amounts of hydrogen. Reactor C produced the most at 12 milliliters, while Reactor D produced the least at almost no milliliters. From these results, we inferred that the differences in hydrogen production yields could be due to the differences in microbial community structures. In order to further analyze the microbial community structures in our reactors, a detrending correspondence analysis, or a DCA, was performed. What a DCA basically relates is how similar or different the community structures of each reactor are to each other. Each dot on the DCA represents samples of each reactor, and the closer the dots are to each other, the more related they are in terms of their community structure. As you can see, each reactor is different from each other. This is consistent with our results because each reactor produced such different amounts of hydrogen. In order to further understand why exactly each reactor produced such different amounts of hydrogen, some key genes involved in electron transport, hydrogen consumption, and methane production were looked at. First, cytochrome genes, or the genes involved in electron transport, were looked at. At the right of this microarray analysis is the name of the cytochrome gene and the name of the bacteria, each having their own row. At the top of the analysis are the name of the reactors, each having their own column. These lines at the top show how similar the reactors are to each other, and the lines at the side show how related the gene and bacteria are to each other in terms of their intensity. As you can see, the color ranges from black to bright red. The level of color intensity signals how prominent that gene and bacteria were in that reactor. This gene and bacteria was prominent in all reactors. From these results, we saw that several metal-reducing bacteria such as Shigella and Geobacter were important for hydrogen production. We also looked at the genes involved in hydrogen consumption, or hydrogenases. Hydrogenases are essentially enzymes that catalyze the oxidation and reduction reactions of hydrogen, and they consume hydrogen. Because many microorganisms in our MECs could use hydrogen as an energy source, hydrogenases could be very important. From this analysis, we saw that Reactor A had most of the hydrogenases, while Reactor C did not. This is consistent with our results because Reactor C was producing the most hydrogen. Finally, we looked at the methane production and oxidation genes in our MECs. Methane is produced as a derivative in our MECs. the microbes in our MECs could have either produced methane or produced hydrogen. But in order to maximize hydrogen production, methane production needs to be limited. We saw that reactor A had most of the methane production and oxidation genes, while reactor C did not. Again, this is consistent with our results because reactor C produced the most hydrogen. Shown here is a canonical correspondence analysis, or a CCA, of our reactors. A CCA relates the performance of each reactor, in this case gas production, to the reactor's community structure. On the CCA are 3 gas arrows labeled methane, hydrogen, and carbon dioxide. The closer the reactor is to the gas arrow, the more related that reactor is to that gas. Reactor C and its replicates are close to the hydrogen arrow. In summary, this experiment is important because it introduced a new process in which clean, efficient hydrogen is produced by using wastewater as inocula. This essentially kills 2 birds with 1 stone. First, clean hydrogen is produced, and second, wastewater is treated. Also in this experiment, my partner and I obtained an effective community of bacteria that efficiently produce hydrogen from wastewater, and we showed that several metal-reducing bacteria such as Zhou and Zhongshi were important for hydrogen production. As a result, MECs could be a powerful approach to producing hydrogen in the future. However, there is still room to improve. Based on the MEC technology, experiments in the future could be set up that could produce even more hydrogen. Also, in this experiment, we obtained an effective community of bacteria that produce hydrogen. However, bacteria isolates can be obtained in the future to narrow down on exactly which individual genes and bacteria produce hydrogen. Stemming from obtaining the bacteria isolates, methane production and oxidation genes can be limited in order to maximize the hydrogen production. Finally, environmental conditions could be understood to understand how exactly environmental changes affect the hydrogen production. If this technology is developed further and spread widely, it could have a very big impact in our society's future. We'd like to thank Dr. Jordi Van Nostrand, who is our mentor, who helped and guided us through our experiment, graduate student Wenzong Liu, who helped us set up the reactors, postdoctor Ye Deng, who helped us with the statistical analysis.
We'd like to thank the judges in the audience for listening to our presentation, the Siemens Foundation and the College Board for organizing the competition. We'd like to thank George Washington University for hosting the competition. We'd like to thank our parents for always supporting us, and we'd like to thank the Institute for Environmental Genomics at the University of Oklahoma, which is where we conducted our research.
Thank you.
Thank you.